Good afternoon and welcome to the St. Croix River Association's Water Resource Forum. This morning, or this afternoon, we have our guest speaker is Jeremy Williamson from the Polk County Land and Water Resource Department. He's been there since 1999. His educational background is a Bachelor's of Science in Biology from the University of Wisconsin, Eau Claire. And he also has a professional certificate in Watershed Management from Michigan State University. He's currently working on water resource projects with Dr. Mark Edlund at the Science Museum of Minnesota and the St. Croix Watershed Research Station. So please join me in welcoming Jeremy Williamson.
All right. Well, for those of you who can't see me and are online, that's me right there in the blue raincoat, pulling a sediment core. So the title of this talk, Beguiled by Big Blake and Bone Lake, I was trying to be clever. I gave a similar presentation earlier at the Research Rendezvous and was trying to match the width of Jim Allmendinger down there. I should also add that Joy Ramsack-Hobbs should be on this as well. She did some of the statistical work on this. But that is us getting ready to push a core. Some of these lakes is going to focus on paleolimnology a little bit. And that is what a successful core looks like. So for those of you who aren't familiar with pulling a sediment core, who may be watching online, it's kind of like if you stick a straw in a milkshake and put your thumb over the top and pull it out. That's just a really big straw. And we pull out about 2 meters of mud. We were just shy there, is 1.9 something, I think. Then from there, we section that core at 1 centimeter intervals or 2 centimeter intervals, depending on what we're looking at. We'll get into the remains and things that we look at in just a bit, and throw them into little cups and analyze them for all kinds of different things. We have to date our cores. If you were to take a core in the Arctic or possibly in the Alpine region, you may see laminations, kind of like tree rings, and you can count them and kind of go back in time that way. Our sediment cores that I've worked on look like mud. So we have to date them. We use lead-210. It's a natural mineral. And generally, we're just looking from pre-settlement to modern times. So we use cesium-137. That's the CS there. To calibrate that, and when it shows up, we know it's 1950. And when it peaks, we know it's 1963, because that's when they were testing nuclear bombs in the atmosphere. But from those cores, we can pull all kinds of different things, which I'll talk about. You got biogeochemical markers. Phosphorus is a big one for lake management, which is what I tend to focus on in my job. We look at different kinds of algae, or indications of algae anyway. Chironomids are some things I've worked on there. Some people call them lake flies. If you've been at the cabin in the summer and you see your windows covered in little things that look like mosquitoes, that's what they are. Sediment characters, all kinds of things like that. You can look at things like magnetics, more magnetics as an indication of more soil erosion through changes in the watershed, be it land use clearance for logging or agriculture or things like that. I'm not going to get too into this. This is a really wordy slide, but you can do almost anything. The science on this, since I've started my career 15 years ago, has advanced leaps and leaps and bounds in the last 2 decades. So the 2 lakes I'm going to focus on are in the red circle. The bigger, longer lake is Bone Lake. The smaller, longer lake is Big Blake. They're at the headwaters of the Apple River, which is important. because the Apple River is the biggest contributor of phosphorus to the St. Croix River, which is why we're representing here. I did add some other dots because we've done some other core work on Horse and Lotus Lake, which is the southern part there. They drain down to Cedar Lake, which also outlets to the Apple River. And Big Round Lake we've worked on, which actually flows to Blake Lake via the Straight River, which of course goes to the Apple River. And we just started a project on North Pike Lake, which goes to Pike Lake, which outlets to the Apple River. So we're getting kind of a comprehensive data set, and I'm kind of excited to move forward. I have some ideas on that maybe I'll touch on. But for now, let's get into what we have. Big Blake Lake, this is a very comprehensive project. As you can see, it can throw some pretty nasty algae blooms. This is in 2011. What's interesting about the blooms on this lake is they come and they go. And they come and they go. It's not persistent. We have other lakes we've worked on where you get a blue-green algae bloom like that, and that's what it looks like the rest of the summer. This is a little different, and I think we're starting to get our finger on it, but talk about that. This is kind of parsing out the sediment. You have inorganic versus organic. So organic would be bits of algae and plant material and things like that that are starting to decompose, and they burn off in a furnace. So to do this, you basically burn off sediment at, I think, 500 degrees Celsius and then 1,000 degrees Celsius. And what's left is the inorganic or mineral component, and that will give you soil erosion, essentially, or bank erosion or stream bank erosion. You can see a big flip-flop right around settlement time. That could be due to land use. But you can see there's other flip-flops. I have no idea how far back in time this is, because we went pretty deep. And it's flip-flop back and forth. But it is part of a bigger river system, so there could be any number of factors that could contribute to that, I guess. But when you do look at sedimentation rates, when you do that loss on ignition and you look at that, you can see that at least since the 50s, sedimentation rates have increased. Phosphorus sedimentation rates has increased. And the flux, or what's being deposited on the sediment, has increased. So it's not surprising. That's what we see a lot. As things change in the 50s, as post-World War II, people have more disposable income. We develop shorelines, agricultural practices change. So we see that a lot. So that's not horribly surprising. But what does that lead to? We know we have algae blooms. So that doesn't really fossilize. Like, I'll talk about diatoms in a moment. Anybody saw Mark Edlund's presentation? He talks about diatoms a lot. The algae I'm interested in, the cyanobacteria blue-green algae, they're soft. So they just break down and decompose. But their pigments, or their colors, are unique, and we can analyze that in the sediment. And what we found there was actually pretty interesting. These are the blue-green algae pigments right here. What you really notice is about a little bit of lag time from the 50s when the sedimentation rate increased. But in the 60s, they really start to come up. And this one, Ephanizophyll, I'm probably saying that wrong, but yeah, that's a definite blue-green algae. It's named after Ephanizomena, which we know is a toxin-producing blue-green algae. And we don't even really see that until the 80s. So when we play with the data, we do some weighted averaging and math, which we won't get into because I'm sure a lot of my viewers aren't really that into weighted averaging and stuff. But you can see these are really grouped tight on these. Except here, I amplified that Ephanizophyll, and they're kind of outliers. So when you pull that out, all of a sudden it's a little more evenly distributed. So you pull that out, things are a little more evenly distributed, and then you look at sediment phosphorus versus algal pigment. And it's the same timing as you see, even without the outlier, about 1960, you see all of a sudden this dramatic increase. Okay? Again, not horribly surprising. We know things start to eutrophy or get green, but I mean, it's pretty distinct. It's a pretty distinct increase. Now, when I add the line here, again, it's the same. Now, all of a sudden, this outlier becomes really interesting because it's not there. And then all of a sudden, in the 80s, outside of the fact that, one, all of the algae pigments are increasing, but the blue-green algal pigments are really increasing. And then you have this pigment here that is not there until the 80s. So statistically, it makes a little bit of a mess when you're doing those things. But now it becomes fairly interesting. So one of the nice things about running these pigments is you get isotopes. You get isotope data back with it. Carbon and nitrogen isotope. Really, all I'm going to say about this is that this carbon-nitrogen ratio becomes important because it's an indication of whether your nutrients are coming from land or from within the lake. And as that decreases, it's likely that it is coming from the lake. Again, we see a lot of internal loading. Big Blake Lake is one of those lakes that's polymictic, means it mixes a lot, so it may stratify for a while and then it'll mix. But likely is coming from within the lake, a big portion of their sediment load. Or not their sediment load, I'm sorry, their phosphorus load. So when you reconstruct the phosphorus using diatoms, these are diatoms, another kind of algae. So you start looking at actual, lake concentration of phosphorus. And if you look at these, the one on the left is Blake Lake in 1852. And the one on the right is Blake Lake in 2012. Without knowing anything about algae or diatoms or phosphorus, you can clearly see that it's a completely different community from 1852 to 2012. So from a reconstruction standpoint, as we're moving forward, it's nice for us because we can see that they're clearly different. When you look at them, these are the 6 most common species here, and planktonic species, which means the ones that are floating around in the water, they're not living on plants or connected to rocks or down in the sediment, has ever increased. What's interesting is this, sorry I don't have the dates over here, there's like a decade indicator here that the diatoms change before we see this big increase in blue-green algae blooms. So if you're familiar with regime shifts or stable states, there's kind of an indication there that things are kind of on the wrong track here. And so if you were continuously monitoring a lake like this, you might start to see these changes over years. I mean, I wouldn't say 1 year that you're changing, but if you're seeing it year after year, you may have a problem, which is not good from a management standpoint. One of the other things we're looking at are zooplankton. This is we're doing multi-proxy stuff here. I am currently working on that. It's very tedious to dig through mud and pick out shells of these little guys. Daphnia, this is Bosemina. They tell us different things. It can tell us a lot about fish predation. Sometimes it can tell us a little bit about plant life, whether there's a lot of macrophytes or not macrophytes. It kind of goes along with everything else we've looked at thus far. One thing that I am finding is pre-settlement. This little spiky thing here with a little wad of hair is a mandible from a Chaabris or a phantom midge. And there's not a lot of those if there's a lot of fish. They get fish predation on them is really high because really all they can do is control their buoyancy. They're predators on other zooplankton. That's the mandible there. You can see it kind of looks like fingers. So they're eating that. They're eating the zooplankton, and they use their antennas as spears. They're kind of cool, actually. There's videos online. I think Oregon State University. So that's an indication. I'm not finding them in more recent samples that the fish community has changed along with the algae. I mean, none of this is horribly surprising. It's the cogs in the system, but you know what's driven the change, I guess, is the big thing. So we move on to some plant macrofossils. Here we have Ceratophyllum demersum, coontail, very common. Everybody kind of knows coontail. You can tell there's a little spike on the bottom there. That's coontail. These are Chara oocytes. Chara is sometimes called musk weed or musk grass. Has a very distinct skunky smell to it when you pick it up. Usually an indication of very clear water. This is Nias. I think it's Nias flexilis. They're a little bushy pondweed. Find that on the shores a lot. This is a seed from a Potamogeton. I think it's immature because there's not a lot of parts on it. And I think this might be wild rice, but I'm not positive on that yet. Now, what I am finding in the macrofossils that I've collected thus far, so we have these distinct changes in the biological community. And, you know, Ceratophyllum is by far, the coontail, by far the most common plant fossil throughout all. It's ubiquitous, it's worldwide, it's everywhere. If you walk down to the river down here, you could probably find some right now. But I am not finding curly leaf pondweed fragments in early samples, and I'm finding that now. So curly leaf pondweed, Potamogeton crispus, it's the only Potamogeton with very distinct crossbands. So you can kind of tell when you have like a piece of grass or an aquatic plant, the grass has more lignin and things like that, it's just thicker, whereas aquatic fossils do not because they're supported by water. And so when you kind of find bigger pieces of the curly leaf pondweed leaves, they're very distinct because there's that distinct crossband across it. So that's fairly interesting. I do know we have been doing plant surveys on the lake since, or I have been since 2006, because they have a lot of curly leaf pondweed. So we're compiling all our existing data. We have Simpson's diversity indexes, and we have some water chemistry now we've been doing for 3 years, and some historic data. Lots and lots of plant data. We're doing phytoplankton or algae work on the lake. So we have a lot of data on this lake. One other thing we've done with it, because generally in lake management, you collect all your data, and here's what we found, and then you go into some watershed modeling. So I worked with UW River Falls. I had a student there work on some historical land use for me. And so I got all of these historical photos, which you can download from UW Wisconsin. I'll say UW Wisconsin. I think it's through the geography department. And you can download historical photos throughout the years. And I had him geo-reference those photos and redo the land use. So with those, we can do some watershed modeling. So we can get some historic loading rates. And eventually, I'm hoping with the more defined phosphorus reconstruction from the diatoms that we just talked about, maybe I'll be able to model out what the internal loading was. Unfortunately, we won't have an annual change in phosphorus, but maybe we'll be able to tease that out and at least have an idea of how the internal load has possibly changed. What's interesting is when you look at the land use, because generally we see land use degrade. Well, when you look at Blake Lake, this is the direct land use. The watershed is bigger, but to do the whole straight river, when all these other lakes have management plans, we stuck to with what this lake group could possibly manage. When you look at it, what I want you to pay attention to is the dark green and the red. That's forest versus developed land. So this is 1938 on the left, 1955 on the right. Obviously, there's more development. Not a shocker there. Post-World War II, people started building cottages. Here you go. You get to 1974, most of the lake is developed, but there's more and more forest. You get to 1996, kind of the same thing. To present day, notice the color of the lake. This is the 2014 aerial photo. There's a lot more forest. There's not a lot of agriculture left in that watershed. So essentially it's kind of been reforested over time. There's a little discrepancy in wetlands. I'm guessing that's because possibly there's some forested wetlands. So I'll have to look at some soils data to define that a little better. But when you look at the changes in land use, you can see it's been reforested, likely because pasture and grass has gone down. This is just these are exaggerated because we only have so much as far as photos go. And the wetlands, like I said, I think there's some forested wetlands we just don't quite have mapped. But when you look at the loading decreased and obviously did not go to zero. That looks like. Then it's increased some. The only thing I see is there's an increase in development. And when I did the modeling for this, I did change the impervious surface ratio on the developed land, because generally those cottages and stuff in the 40s were 5 to 10% impervious, whereas we've actually gone to several lots in lake studies that we've done and measured, corner to corner, from driveway to house to whatever, and on average we're at about 21% impervious. There's more and less, but on average it's about 21%. So I did change that. So from the land use standpoint, our loading's decreased. It's up a little bit, again, since the 70s, it's increased, and that's likely due to shoreline development with the increased impervious. But in general, it's less than the 30s. So as we connect the dots here, let's go back. As we connect the dots, it's looking more and more like our management strategy is kind of what we're doing right now. So we're promoting getting rid of curly leaf pondweed. They've chosen to use an aquatic plant harvester. So they're also removing the biomass and the nutrients with that. And the macrophyte or plant studies that we've done has showed a decrease in density. So I think this will be an ongoing project as they keep removing more and more curly leaf to see if that approach is going to work. There is, if you go on the lake, there is some work to do in the riparian area on the shorelines to catch some impervious surface. But that's the nice thing about that is that's something we can control. People can do that themselves, and it's not going to be a huge cost of doing barnyards and manure pits and things like that. So that's the upside. The downside is it's very hard to control curly leaf pondweed. So on to Bone Lake. If you recall, Lake had a big change in inorganic and organic sediment contact. Bone Lake hasn't changed very much. Kind of straight up and down. So what does that tell us? I mean, you can basically spit to Bone Lake from Lake. They're both connected via the Fox Creek. They have the similar orientation, north-south, long, skinny lakes. They're both mixed. Bone Lake's a little deeper. It does stratify longer. Sometimes it will stratify all summer long. Sometimes it won't. So it's right at that cusp. It's about 10 meters. So it can mix, and sometimes it stays stratified. So what's going on in Bone Lake? Well, sedimentation rate up twofold since pre-settlement. Not surprising. Changes in land use, land clearance, shoreline development, agriculture. There's not a lot of surprises there. On this one, I have the data. We have the phosphorus fractions. So the green line, which is kind of hard to see, and the orange line That's organic phosphorus, and phosphorus that's bound to manganese and iron. So in the top 4 centimeters of sediment, that's where it's built up. Not surprising, phosphorus is mobile in sediment, it'll move. So if the lake does stratify, and we get a mixing event, say midsummer, if it's anoxic, that green and orange sediment, or phosphorus fractions in the sediment, could be available to algae and spur algae blooms. This past summer, for example, they had a huge blue-green algae bloom in June. Usually it happens in the fall, but they had one in June this year. And killer waterets, apparently, according to the folks I talked to on the way. So I had the phosphorus fractions. They are doing an internal loading study right now. It's very intensive, and they have dozens and dozens and dozens of points that they're actually testing. Some of the flat, we'll call them bays. There's not really bays on the lake, but some of the flat areas, the flats on the in the riparian area, or not the riparian, sorry, the littoral area, they are doing some internal loading, because if it does stratify, there's probably still loading from those shallower areas. What's really interesting in Bone Lake is the biogenic silica content. That's silica, used to make glass, generally used by diatoms. The biogenic silica content in the sediment is 24 to 44% of the sediment weight, the inorganic portion of the sediment. Most lakes is 2 to 6%. So this is actually higher silica content than Lake Baikal in Russia, in Siberia. So it's really high. This may be throwing some of our conclusions off, is because there's such a high content of this biogenic silica. Because I'm going to go back. You look at the organic versus inorganic. It hasn't changed a lot. But when you look at the organics and you take out the biogenic silica, so essentially the weight of the diatoms. Oops, wrong button. It's decreasing. So Bone Lake has done a lot of work. They've done a lot of shoreline restorations. They've done some sediment basins. They've done a lot of work. So it appears as though what they're doing in the watershed is reducing their sedimentation. They just have a lot of silica. And again, when you look at the diatoms on Bone Lake, it doesn't quite look the same as Blake. They're kind of the same. Obviously, similar species, right? But just looking here, there's a lot more. And that goes again with the biogenic silica. There's a lot more. And when you look here, there's now all of a sudden an increase since about the 60s in biogenic silica. How I would interpret that is, because there's so much silica available, is that Bone Lake is increasing in productivity, even though the sedimentation rate has increased a bit. The soil erosion has increased. And this is one screenshot from a microscope slide. But it certainly appears as though that kind of seems to be the case. And they are experiencing blue-green algae blooms periodically. It's not persistent, it's not all the time, but they are concerned about it for this project. So when you take the most common species of Bone Lake's diatoms, this Alcoceria, that's a mesotrophic species. Most of these are typical of a mesotrophic eutrophic lake, going way back in time. There's 110 total species found, but these are the most common. You see there's a decrease here, this Alcoceria, I hope I'm saying that right. That's kind of a mesotrophic species. You find it in a lot of mesotrophic lakes. Fragillaria cotinensis, there's an increase. That's a eutrophic species. A lot of these are what they call centric diatoms. So they're these round ones. They're floating around in the water column, absorbing nutrients from the water column. that's how we reconstruct phosphorus. It's based off of whether they're floating around, the planktonic ones versus the benthic ones or epiphytic ones that may be growing on plants or in the sediment. So this looks like a lot of math. But when you look at these, again, it goes back to, it doesn't look like bone lakes changed that much. And most of the change is from 1900 to 1980. Shocker right there. But it appears as though when you cluster it up, the last couple of decades, it seems like it's changed. We're not quite sure how. Like maybe there's multiple stressors, because Bone Lake also has curly leaf pondweed. It could be more mixing, more intensive development. I mean, who knows for sure. I'm hoping to coach some undergraduate student at UW River Falls to redo land use for Bone Lake as well. So there's not really a lot of clustering even. They're kind of all over. And that, we're not sure what to say about that, other than the fact that Bone Lake is probably kind of walking that knife's edge. They're ready for a change one way or the other, but it's going to depend on management. And I threw this up. This is Bone Lake. These are our sections over the top of the Minnesota Lake training set. So training set is they take surface sediment from, I forget how many lakes they had, but a lot of different lakes throughout Minnesota and different ecoregions. We throw Bone Lake over the top, and it's the ecoregion that we're in, so it fits right in there. We do the phosphorus reconstruction. Again, it doesn't look like a lot of change. There's an increase, and it models out as a little bit of a decrease, but that's modern, so who knows if there's sediment mixing from midges or anything like that. But again, what we think is that Bone Lake is kind of walking that edge of they could tip one way or the other. So they are also doing some phytoplankton monitoring. I have volunteers out there taking algae samples every month. They're doing water chemistry through the Citizen Lake Monitoring Program. And so we're getting a handle on it. They kind of know they're walking the knife's edge. They're doing that internal load study. They're doing a lot. And luckily we have great volunteers on a lot of the lakes that we work on. And then I just threw this up because we just started North Pike Lake. The pigments for Bone Lake will be back. I did forget to mention that. We are doing pigments on that lake too. Because we didn't see huge changes in the diatoms or anything, perhaps we'll see a big change in the pigments, because it was pretty drastic on Blake Lake. North Pike Lake seems a little bit similar to Bone. There's not huge changes. The top little bit here, it looks like we're getting closer to organic versus inorganic, so there may be more some autochthonous production or in-lake production of algae. We did see quite a bit of algae production this summer. We're doing similar to what we did on Blake with North Pike. We're doing in-lake water chemistry, we're doing phytoplankton and all of that. But what we really, I think we have a real opportunity. I'm gonna go back, sorry for you YouTube viewers that have to watch me go back on this. I'm going to go back to my original slide of the Polk County map. Because this, there it is. This is where I think this has the potential to really kind of change management, not just on a lake standpoint, but possibly on a larger river standpoint. When you look at Bone Lake and Blake Lake, the Big Round, which flows to Blake, the Pike, which flows out here to the Apple River, and these down here, which flow here, ends up in the Apple River in the St. Croix. We're talking with Balsam, and that Balsam branch also ends up in the Apple River. So the question is looking at the biogenic silica content, looking at, don't want lakes to look like that, looking at the changes in pigments, things like that. Bone Lake seems to have the potential to bury phosphorus. So if it's bound with calcium, so if you have a lot of plant life, so if you change the plants in Blake Lake, water runs out of CO2 very quickly in the lake. Online viewers are going nuts right now, and I apologize for that. But so they tend to use bicarbonate as a carbon source for photosynthesis. So now you have free calcium floating around, and phosphorus will bind to that and form apatite. And apatite is very redoxin sensitive, so you have a potential to bury that in the sediment. So in these river systems, these are all drainage lakes that I'm working on. So in these river systems that flow through these smaller lakes, if we restore the functions of the lakes, not just look at the watershed that's flowing through the river, but that the river flows through. You know, like you might take a SWOT model, for example, and look at crop rotations and tillage practices. But if we can find a way to better model and tease out what these lakes are capable of, perhaps you can use them as a mechanism to actually remove phosphorus from the bigger watershed, as long as they're functioning properly. You know, we have one that we've done cores on that's full of carp. Well, we got to get the carp out. We're hoping to do that today. We'll be wrestling the carp later this afternoon, but I don't think that's going to happen. You know, restoring the native plant community. So you have those plants, and you're oxygenating the sediment with those, so you're not releasing phosphorus, but also you have the potential to bury it. If you've been swimming in a lake with a lot of plants, you notice that film on them, the marl, that's calcium. Particularly para has a huge potential to throw out a lot of calcium radicals. So if we look at the lakes as a bigger part of the river system and focus some more attention on restoring those small lakes, on top of course, you have to shut off the tap, you have to do watershed practices. But as another mechanism of river management, you know, possibly to get our 20%, because I think we did some back of the envelope calculations in our office for the phosphorus reduction Polk County has to do to meet the St. Croix River watershed nutrient reduction goal of 20%. And we need 14 more staff and $50 million to do it based off of current costs of best management practices. So I guess with that, I would entertain questions from the small group we have here. Online folks can't do it, but
Can you go back a slide, please?
one slide.
Yeah. How do you explain those reciprocal spikes outwards?
I don't know yet. I don't have enough data to look at it. I just got this the other day. It could simply be part of the error bar. Sometimes these have pretty decent sized error bars on them.
And does Fox Creek run from Lake to Bone, or
They both outlet to Fox Creek. So Bone Lake has Procop Creek and a couple other creeks flowing into it. And then it outlets to Fox Creek. It's almost to Blake, and then Blake outlets to Fox Creek, and that goes down. So the Straight River, the big state park, Straight Lake, Straight River, flows down through Little Blake Lake into Blake Lake, and then out to Fox Creek. So, you know, yeah, with this this state park, should be natural, shouldn't be contributing a lot of phosphorus. But whereas like Lotus and Horse Lake, which I didn't present on, I think they're a net contributor of phosphorus to Cedar Lake, which goes to the Apple River because they're full of carp. And we're working on that right now. I have automated samplers out there. Things like that.
Horse Lake has always been full of carp.
Yep.
So is that just the life cycle and the dying of the carp? Is that what's...
It's their feeding habits. They stir up the sediment and rip out all the macrophytes. It's a pretty shallow lake. Those two lakes tend to have fairly small carp. So I think there's just a lot of them. I've done sampling on the outlet of Lotus Lake where I couldn't even take a flow sample because there was so much fish biomass I couldn't get my wading rod in there. And I think I've helped a little bit, but the DNR and St. Croix Tribe, they've marked well over 3,000 fish out of Lotus Lake. And we have radio tags in some. One of them right now is hanging out in the wetlands in between Lotus and Horse Lake. So, but we are going to try and remove them. with that. So, and if the macrophytes come back there, then hopefully those lakes won't be a source of phosphorus. They'll hopefully pull it out.
Okay, thank you. Thank you, Jeremy. Our next forum will be on January 28th, and I believe Byron Carnes?
21st.
21st, I'm sorry, that's correct. January 21st. Byron Carnes will be here to talk about his work at the National Park Service. So please join us then at noon on the January 21st. Thank you.
All right.